we have any questions? Eight classes left.
Wait, total?
Total.
That doesn't mean
Three this week, 2 days, 3 last week.
Oh. You're not counting that lab.
No, 3, 8 lectures left.
8 lectures.
How many points do you have left in class, like curiosity?
Lot.
Lab exam 100, final 100,
Unknown.
unknown 150. I did your integral tube, 30 for the flowchart thing.
How much is the ID?
ID was 10.
Did we already submit
You already said that?
a little, like, we need to, like, turn into, like, what computer we're on or whatever. When is that happening? In lab next week?
You've got to have your one required one done by Wednesday. Like I said.
And that's when we're talking.
Okay, we're still on line 2. We're a little bit behind, but we'll do some catching up today. We're going to talk about the complement system and inflammation. Had anybody ever heard of the complement system before? Where did you hear about the complement system before, Anna? You don't know? It sounds familiar. Okay. This is a fairly complicated system that consists of 26 serum proteins that interact with one another when infection occurs. And there are some things that always are an outcome of the complement system, but there are other things that may be an outcome depending upon how the activation of the system took place. We'll talk about that just a little bit more in a second. Where do these proteins come from? Well, liver cells can produce them. Monocytes / macrophages can produce them. And so can lymphocytes. These react with one another in a fashion that we refer to as a cascade. And a cascade occurs when something is initiated, and then that stimulates something else, that stimulates something else. It's kind of like the rock at the top of the hill that rolls down and hits a couple of other smaller rocks, and then they go off and hit smaller rocks. And so we get really an amplified response. So this really is a positive feedback response. So it really amps up. What's its goal? Well, the goal of the complement system is varied depending upon the initiation, but in general, one of the major goals is to lyse pathogens. And the way that that happens is that basically the complement system results in holes being punched in the cell membrane. And that occurs when something called an attack complex is formed. So I'll show you in just a second. Additionally, some of the byproducts of the complement cascade can stimulate chemotaxis that will attract white blood cells to the site of infection. So this would be a positive chemotaxis directed at leukocytes or white blood cells. And then the other thing that really is important in most complement cascades is the cascade can send out signals which will stimulate inflammation. I know we haven't talked about inflammation yet, but that's next on the agenda. If you want to get an idea of how this cascade occurs, take a look at this slide for a second. And it's certainly not something that I would expect you to memorize. If you were taking an advanced microbiology course, an immunology course, then you probably would have to know all of these complement cascade sequences and which particular components reacted with each other ones to then activate another sequence. It gets pretty complicated. But you can see over here that one of the results of the complement cascade is the formation of all of these little attack complexes on the surface of the cell. So certain of these serum proteins, or parts of them, come together and form this structure, the attack complex, which basically punches holes in the membrane of the cell. This works better for gram-negative bacteria. The gram-positive bacteria with their thick peptidoglycan layer, it's a little bit tougher for these attack complexes to form. But additionally, along the way, as this cascade is occurring, some byproducts are spun off, some kind of waste products, if you like, almost. They're not used to make the attack complex, but they have other important functions. So, for example, there are situations where some of these are causing inflammation. Okay, so this little C4A right here is something that stimulates inflammation. Here's a C5A component, which is chemotactic. Here is something down here, I can't even read the numbers, C3 something, which is an opsonate. Okay, attaches to the cell membrane of a pathogen and it enhances its phagocytosis. So there are a lot of protective functions associated with this complement system. Now, there are actually 3 different complement pathways that occur. And they are referred to as the classical pathway, the alternative pathway, and the lectin pathway. You might see this on old exams. The alternate pathway is also sometimes referred to as the properidin pathway. So those two terms are synonymous, alternate and properidin. All 3 pathways merge at the same point. And the same point that they merge at is the formation of this attack complex. As those pathways are funneling toward that point, some of them produce opsonins, some of them produce inflammatory compounds, some of them produce chemotactic compounds. That's all you really have to know about the cascade. I'm not going to ask you to know any of the specifics about the cascade. So what's the difference? The difference is the initiator. Okay? In the classical pathway, the initiator of the complement cascade is binding of an antibody to an antigen on a pathogen. Where's that antibody going to come from? It's going to have been produced by a B lymphocyte. This is one of the ways in which antibodies can trigger pathogen destruction, by activating the complement system. Most people, when they think about antibodies, have a misconception about how they work. Most lay people, if you tell them about antibodies, they have this image in their mind of these things that are kind of like little assassins. And that the antibody itself attacks the pathogen and kills it. Has anybody ever seen the cheesy old 1960s or 1970s sci-fi movie called Fantastic Voyage? Somebody's got a blood clot and they miniaturize a medical team with the submarine and inject it in the body. And they move around through the circulatory system until they get to the clot and the glass of lasers. You should check it out sometimes. It's pretty cheesy. There's one scene where one of the characters has to go out into the circulatory system to repair the vessel. And they start getting attacked by antibodies. And these antibodies are seen like these kind of almost ropes that go around them and start strangling them and things. That's not how antibodies kill cells. That's not how they inactivate pathogens. This is one of the primary ways that they inactivate pathogens, by binding to their surface and then initiating that complement cascade, which eventually results in the formation of that attack complex. You all know that mismatched blood is a problem in a transfusion, right? But you know why? It's because of the complement system, okay? If you have antibodies that react with the transfused blood, okay, so if you get type B blood and you've got B antibodies in your circulatory system, that activates the attack complex and lyses that red blood cell. You die from that transfusion reaction because of the red blood cell lysis. The alternative pathway does not depend upon the formation of an antibody complex. Instead, molecules typically associated with pathogens can interact with one of those 26 complement proteins that the liver or the monocytes or the lymphocytes are producing routinely. They're circulating around in your blood. And in this case, it's C3b. You don't have to remember that. But C3b interacts with an endotoxin or a glycoprotein on the surface of the pathogen. And that complex is the initiating factor that gets the cascade going. It goes in a slightly different direction than the classical pathway, but it still results in the formation of attack complex and typically some of these inflammatory compounds, chemotactic compounds, etc. A third pathway, the lectin pathway, is activated by mannose molecules. Mannoses are sugars. And they are typically not found on the surface of human cells. Mannose, it's unusual to find that sugar as part of the glycoproteins of human cells. But it's not uncommon at all for mannose to be in the glycoproteins of a pathogen. There are substances in the body called lectins which selectively bind sugar molecules. And there are mannose lectins. These lectins are also floating around in your blood. So if a mannose binding lectin sees a mannose on the surface of a pathogen, it binds. And that then stimulates a slightly different pathway of the complement system. And again, you get to the same place, but the direction that you go when the intermediates are produced are a little bit different.
So you know how like some people have like autoimmune diseases? Can that be caused by mannose being on human cells rather than?
There are other things that are more typically going to cause autoimmune disease, and time permitting, we'll talk a little bit about autoimmunity. But no, this is something really different. This is kind of a pathogen specific. Okay. In autoimmunity, Hannah, what really happens is immune cells that react with your body cells were not eliminated properly during the education of the immune cells. And we'll talk about that more in detail. So this one, again, is just a little bit simpler demonstration of what we're talking about. So in this particular case, here's a pathogen with some antigens on its surface. Here are some antibodies that bind to those antigens, so this would be the classical pathway. Once the antibodies are bound, then complement proteins are activated. The attack complex then is formed as those proteins interact with one another, and we form this channel in the membrane, which results in cell lysis. And here's a cell with multiple attack complexes on its surface. And the cell is a goner because of the activation of the complement system. This is non-specific, okay? There's a lot of different things that have triggered this particular pathway. So it is considered to still be part of the second line of immunity. It is not the specific adaptive immunity that we'll be talking about in a little bit. Another very important part of the second line of defense is inflammation. It's important to understand that inflammation is part of your body's defense systems. But are there side effects associated with inflammation that are not pleasant? Absolutely. And as a matter of fact, we're now finding out that states of chronic inflammation, as opposed to acute inflammation. Acute is something that happens, it doesn't last very long, and the inflammation subsides. But states of chronic inflammation actually can be damaging rather than protecting. And that is definitely the case in autoimmunity. Anybody ever seen or know of someone that has rheumatoid arthritis? Yeah, the joints become incredibly painfully swollen. You become really immobile in those particular joints. And that is the result of immune cells responding against host antigens. It's an inappropriate response. And part of that response is inflammation. And the inflammation can actually result in tissue destruction, swelling, and all of these things that are really intended to be protective, but in this particular case, the chronic state is not. We're also finding out more and more that we think that chronic inflammation might play a very important role in coronary artery disease, and particularly in the formation of plaques. What has been the common wisdom as to what primarily initiates the formation of plaques? Cholesterol, bad cholesterol, okay, LDL. But now we're thinking that the initiating events might actually be inflammation reactions. Anybody ever heard of the C-reactive protein test? CRP? That's a test that is intended to measure your body's state of inflammation. And there are a growing number of physicians who believe that a CRP test might actually be more predictive of potential for coronary artery disease than cholesterol tests. But inflammation might play a much more important role than actually these bad cholesterols do. Even some of the bad cholesterols, there's some suggestion that the reason they're bad is that they stimulate inflammation. So let's dig into this process a little bit. See what it is. What's it intended to do? How does it work? Anybody know anything about inflammation? You've all experienced it, I can guarantee it. Throw some things out.
Triggered by some kind of stimuli, which is N-mastol, which releases histamines, which cause the vasodilation process.
Okay, hold on. Okay, so there is typically an initiating event, which is some type of injury. That injury could be a burn. It could be a mechanical injury, like a twisting of an ankle. It could be a cut. Are these injuries always associated with pathogen entrance into the damaged tissue? No. Okay. If you twist an ankle or something, that doesn't expose your tissues to pathogen. Now, if you have a compound fracture and break a bone, that's another story. Okay? But it does not have to be pathogen associated. What were you going to say, Andrew?
Oh, I just said, when you have inflammation, you increase.
Okay. There are certain signs that are associated with inflammation. And this has been observed for a long time in scientific writings. These are the Latin names of the 4 classical signs of inflammation: rubor, tumor, calor, and dolor. Anybody know what any of those 4 terms mean?
Calor is the more.
Calor is the heat, yep. You know what rubor means?
That's swelling.
Tumor is swelling, good. Got a couple more. Rubor is redness. And dolor is pain. When I've had athletic training people in this class, they have said, we usually add a 5th sign to this, which is typically loss of function. That's part of the purpose of inflammation, is to give your body a good signal that you've done something to it. And in many cases, these particular symptoms result in you backing off and particularly not continuing to do the things that caused the injury in the first place. What kind of things can counteract these symptoms of inflammations? Don't know about that one. I don't think adrenaline is. You mentioned one already. Pardon me? Antihistamines, because there's some chemical compounds called histamines, which are associated with inflammatory responses. Basically, allergies are inappropriate inflammation responses. What do they sometimes do for athletes who are suffering severe inflammation? Try to bring it up. Inject steroids, yep. Cortisones and things like that. And that will cause the symptoms to subside, but is that a good thing? No. I mean, because loss of function is supposed to be part of the process, okay? So if they inject you with something that causes the swelling to subside, the structural damage might still be there, and probably is still there, and therefore you run the risk of further damaging yourself. Pardon me? Ultrasound in what context?
Not the imaging, but I get a ultrasound.
Oh, for, okay. Ask your person who's giving that to you what it's doing. I'm not familiar with
The
it.
wavelengths are breaking up the scar tissue and the inflammation line.
Okay, I can understand the scar tissue. I'm not sure what's doing for inflammation, but
I don't either.
yeah,
Same.
good question. I'll have to ask them. Okay, so why do these things happen, especially the first 3? What causes the redness? Vasodilation, bringing more red blood cells to the site of damage. And red blood cells are red. And so that increased blood flow is the result of redness. What causes the swelling?
The water leaking out of the blood vessels.
In part, which is due to vasodilation. Okay, those junctions between the endothelial cells get a little bit sloppier, increasing the ability of those white blood cells to crawl out of the circulatory system into the tissue. And when the white blood cells crawl out, fluids can leak out again too. Where does that fluid eventually end up? Why does your swelling go down? Yeah, it's absorbed by the lymphatic system, and any pathogens that might have been introduced are carried along with that lymph into the lymph nodes. And what do they encounter there? Immune cells. Okay. Should we kill them? A lot of T lymphocytes particularly. So really, the goal of inflammation is to increase blood flow to the area. Bring more protective cells. These cells are metabolizing. Remember that the phagocytic cells undergo a phenomenon called a respiratory burst. Increase oxygen utilization, therefore start generating all of those ROSs, reactive oxygen species, and their lysosomes. That's part of the killing process. So as metabolic rate is increasing, what's going to happen? What's the waste product produced in metabolism? CO2 from this list here. Heat. Okay. So the more cells you bring there, the greater the metabolic activity, the more heat that's going to be produced.
You said the metabolic activity occurs because of more cells.
No, metabolic activity occurs to results from phagocytosis. But then the more phagocytic cells there are, the more overall net metabolic activity there will be. So it's really a product of more cells and that respiratory burst. The leakage of fluids also has some protective function as well. If there are pathogens and they are producing toxins, that will help to dilute the toxins. Blood clotting proteins can start to leak out into the tissues where the damage has occurred and start forming clots. Remember that the clots have a couple of functions. They can both start sealing off areas where there have been ruptures in a vessel or the skin, but they also tend to wall off and contain the pathogens as well. So forming clots is good.
I'm just with Dr. Mann right now, and we looked at contractors a lot of the vein disease, and I know those veins are quite large and stretched, that's why they don't work as well. And he talks about like DVTs and stuff in the tissues and walling off things. Is that due to the veins being more leaky?
That would be my guess, yes. The more clotting proteins you're getting out there. Yep. Yep. These protective cells that are getting out of there are phagocytic cells. Later on, they might be macrophages / dendritic cells. And eventually, those macrophages or dendritic cells might end up recruiting lymphocytes. So antibodies start being brought to the fray. Damaged tissues can start pouring out chemotactic substances to attract the white blood cells. As I use the word cytokines. Damaged tissues can also produce cytokines. Those are chemical signaling molecules. And a lot of those cytokines increase vasodilation and therefore increase vascular permeability. So there's a lot of interesting stuff going on here in this process of inflammation.
What did you say about cytokines?
Damaged tissues can actually, when they're, tissues damaged during the trauma, can release these cytokine compounds that starts increasing vascular permeability, like histamines, for example. Okay, so here's inflammation, here's our initiating injury, and there are the signs of inflammation. Oftentimes the pain itself is what results in the loss of function, although the swelling contributes to that as well. But again, remember, this isn't intended to be damaging to you, it's really intended to be a protective process. Okay, this one's a little bit hard to see. I'll show several other slides which kind of show the same thing. So here's pathogens that are entered into the body by, in this case, a splinter. Okay. The damaged tissue, and in some cases the pathogens themselves, can release cytokines, which are chemical signals. And some of those cells that are confined to the circulatory system eventually can then leak out of that circulatory system as the permeability of the vessels increases, as vasodilation occurs. There's some interesting things that are going on with cytokines that stimulate both the endothelial cells and the neutrophils in this case. Endothelial cells start producing molecules called adhesins, which white blood cells can bind to and kind of stops them from rolling along the surface of the cell. And that's one of the first steps in that cell creeping out into the tissues. The white blood cells start producing selectin molecules, and the selectin molecules bind to the adhesins. I don't know if you remember from that Inner Life of the Cell movie, but you've got those white blood cells rolling through the circulatory system, and at some point they kind of come to a stop and they seem to stick to the cell wall surface. And then they eventually move into the tissue through the junctions between those endothelial cells. So the goal is to get some of these protective cells out at the site of infection, and then as the fluids leak out, that brings other things into play as well, various chemotactic substances. Okay, so here's normal permeability of the venule. Very difficult for a monocyte in this particular case to squeeze out between those cellular junctions right here. Small amount of fluid leaks out, that's inevitable, you're always going to get that. But then under the influence of mediators of inflammation, histamines are a big one. I'll mention another one called bradykinin. That's actually a byproduct of the complicated blood clotting process. Bradykinin spun off during the clotting cascade. Some of the complement byproducts might be initiating this vascular permeability to increase. So now, the monocyte or the neutrophil or whatever starts squeezing out into the tissues. Now we're getting some protective cells there. And then more fluids leak out at the same time. So we're going to get some of that swelling. Swelling can press on nerve endings and result in some of the pain. This compound, bradykinin, and another compound that's important in inflammation, prostaglandin, really irritates nerve cells and causes much of the pain.
Why is it, like, I understand if you, like, spring fever or something, like, the swelling would be important, like, it would be important to keep you off of it, like, so that you feel like that makes sense, but, like, if you were to get, like, a splinter, like, what is the big deal of, like...
Because infectious agents could have been introduced with that splinter.
Right, but, like, what's the importance of, like, your finger, like, having pain, like, staying, you know what I mean? Like,
okay. Yeah, well, I think that's a good question, Hannah, and it really gets back to the nature of the response. And it is a second line generalized response, and so it's kind of a one response fits all.
Okay.
So in
it's
some
not
cases,
necessarily
the
important.
pain would not be appropriate or even needed, but that's just a byproduct of how the general system works. Okay?
Is it prostaglandin skin oil?
Pardon me?
Is it prostaglandin skin oil?
Skin oil. I don't believe so. Okay, this is just another look kind of at the same thing. So here is a cut penetration of the epidermis and some bacteria introduced. We've got damaged cells releasing these compounds that are the cytokines that are really stimulating some of the inflammation. Prostaglandins, another one called leukotrienes, you might have heard of that on some ads on TV. They're involved in asthma as well. Histamines right here. Okay. These compounds, prostaglandins, leukotrienes, make the vessels more permeable. So do histamines. So now macrophages and neutrophils start moving out into the surrounding tissue. Clotting proteins start moving out in there, and we start getting some clotting here. We start getting some initiation of tissue repair. We've got macrophages kind of engulfing some of the damaged tissue and cleaning it up. We get swelling causing nerve pain, and also things like these prostaglandins contributing to nerve pain as well. And eventually this particular process is intended to be self-limiting, and ultimately the threat should be resolved, the healing should begin, and eventually as the tissue is repaired and we stop producing these inflammatory compounds, inflammation subsides, blood vessels start to constrict again, things don't leak out as much, and eventually you're back to normal. And then this is just kind of a flow diagram that kind of shows you the same thing. Some people like this way of understanding it better than others. It's really the same idea as what we're showing right here. So these are some of the major chemical mediators of inflammation. Vasodilating chemicals, chemotactic chemicals, and then there are some that have both vasodilating and chemotactic functions. So you see here, histamines, bradykinin, prostaglandins, fibrin, which is one of the blood clotting proteins, mast cell chemotactic factors, bacterial peptides, down here, complement fragments, interleukins, leukotrienes. There's a whole bunch of different compounds that are involved in the process of inflammation. So, we've got a set of drugs which are somewhat useful in limiting and inhibiting inflammation. Okay.
So, I may have missed this, but why is it beneficial for our body to have inflammation response to fracture?
It really gets back to what Hannah said. It's a generalized response to kind of damage, and the response has built in adaptively things that would take care of pathogens if they got in, but the same response is triggered when pathogens aren't introduced. So the body just developed this generalized mechanism of dealing with trauma, and it kind of covers all the bases. Okay? Some of the things that can inhibit inflammation include some of the compounds like antihistamines. And there are a whole battery of other kinds of treatments that you can use for treating inflammation associated with allergy. I don't think we're going to get a chance to talk about those, but there are a whole bunch of allergy medications out there. One of the problems with histamines is they make you drowsy. And some of these other compounds, like Claritin, for example, don't make you as drowsy. But they're all intended to inhibit inflammation. And then steroids will inhibit inflammation. But steroids do that by generally depressing white blood cell function. So that inhibits immunity as well, which is problematic. And then we've got this class of drug called the NSAIDs. What do NSAIDs stand for? non-steroid anti-inflammatory drugs. And they include things like aspirin and ibuprofen. You may know how the NSAIDs work. Yes?
Inhibit
They inhibit, I don't think all NSAIDs inhibit COX-2, but ultimately the NSAIDs inhibit the formation of prostaglandin. And there is a class of them called COX-2 that is a particular enzyme in the pathway of forming prostaglandins, called the COX-2 inhibitors. But if you take these drugs, then it decreases inflammation by increasing prostaglandin, and particularly decreases pain. What else do steroids or NSAIDs decrease besides inflammation? Pain, which is part of the inflammation process, but you said it, right? Fever. Okay. So the last thing we want to talk about in line 2, which is kind of another generalized response, is fever. Typically, the purpose of fever is to augment inflammation or other immune responses. Help. Yep. Reinforce. There are chemical compounds called pyrogens which initiate the fever process. These are the stimulators of fever. Those pyrogens can either be endogenous or exogenous. Exogenous pyrogens would be things associated with the pathogen itself. So that there are molecules associated with the pathogen that can trigger fever. Endogenous pyrogens are produced by your immune cells or your immune system. And they could include things like complement proteins that have been activated, or various cytokines that are produced by your white blood cells. What do these pyrogens do? Well, here's the common wisdom right now, what they do. They travel through the blood, and they affect the hypothalamus and cause the hypothalamus to produce prostaglandin. There's that prostaglandin. Okay. And what does prostaglandin do? It resets your thermostat in the hypothalamus. And it raises it. And that has the same effect as turning up the thermostat in your room. The heat kicks on again, and the temperature rises. And as long as that thermostat remains set high, the body temperature will be higher. Well, what do you think the possible benefits are of having this fever? Okay, it could denature bacterial proteins and kill cells. The risk being that kills your cells too. So at some point in time, you really do want to treat fever if it's going to raise to the point where it starts killing your cells. And if you're above about 104 you better start trying to bring your fever down, huh?
Vasodilation.
Potentially, okay, could stimulate some vasodilation, yep, as a response to increasing temperature. Actually, vasodilation is oftentimes more of a byproduct of fever, because what your body's trying to do is it's trying to bring more blood to the skin surface, and so you can radiate away that heat to protect body core. Okay, so that's why redness is oftentimes associated with someone who has fever. Little kids spike fevers really fast sometimes, so they just get bright red, really. Fever, it appears, increases the activity of immune cells. For example, interferon seems to be more effective at a higher temperature. Another thing that temperature seems to affect is iron availability. When you get more feverish, the body stops releasing iron stores into the circulatory system. And remember, pathogens really need iron for their growth, so that can help you too.
I know you said fever activates or increases activity in immune cells, but it also, in a way, does that mean like it increases their metabolism? Because I know they're one of the byproducts of metabolism.
It will increase their metabolism, but it really also actually affects their production of immune cytokines. There are environmental factors that can influence gene expression. And in immune cells, one of them is temperature. So they actually start expressing genes for these immune proteins more frequently. So as your temperature starts rising, vasodilation occurs, okay? Sweating occurs. And eventually the fever seems to kind of reach a crisis. And oftentimes that's really associated with very profuse sweating. And then as the immune system really eliminates the threat, then the cytokines that stimulated the prostaglandin release by the hypothalamus start to decrease. Prostaglandin levels start to drop, and now the thermostat drops back down and resets itself to normal values. So what do the NSAIDs do? By inhibiting prostaglandin synthesis, they then effectively help to lower that thermostat. So, should you routinely treat a fever? You really shouldn't, okay? Because you are compromising one of your body's then defense systems. But you've got to monitor it and be careful. If it gets too high, then obviously the negative effects of the fever outweigh the benefits. But if you can tolerate a low-grade fever, it's really better to let that fever go and let it do the benefits that it does. Okay, so we're finally ready to move into the third line. So what we're going to do tomorrow is just refresh a little bit on the third line and then talk about some essential ideas that are important to understand the third line. And they have to do with some understanding of some cell surface receptors, what are antibodies, what are antigens, and then we'll finally put it together into a coordinated response. All of the notes for this have been updated, and they are on the website in an updated form. I will go ahead and send out the notes and the PowerPoints in case you have difficulty getting to those for some reason. But basically, there's one big PowerPoint, and there's one big set of notes for the third line of immunity.
And study guide.
Yeah, study guides are on the website too. I can send study guides.
Yeah.
Send it on.